This is a revolutionary approach in pharma for drug dynamization. We have been working in this field for more than 30 years in this particular field. The information about presenter. And briefly, why we start these researches. One fragment of my education, I read in childhood a book, Da Vinci, and he wrote that human food is a masterpiece of engineering and a work of art. I was very curious about this because he never mentioned Mona Lisa, but mentioned food. Finally, I got to a few postgraduate school. One of them was the leading institute in this domain, Central Institute of Prosthetics and Prosthetic Design in Orthotics. This is our institute. And I decided to start studying this food. First of all, I got a lot of information. I was very surprised that majority of computation was based not different wars, not different tumors, but almost 82% is neuropathy and vascular disease. So the best food, ideal situation when you don't have any food, not any prosthetics. And I started getting information about how to decrease number of amputations. After that, when I got some information, I visited Henry Saulowich Alshuler and share my thoughts that in parallel to creating artificial feet, we're supposed to also think about how to prevent these amputations. He approved this idea, and after that, I wrote open letter to my colleague, to friends, and basic idea of this, that today we are doctors, and tomorrow we are patients. You can't avoid this very important field like medical drugs and pharmacy. So I sent this email a few times in different years and created a team. So in parallel, in this slide, they represented inheritance of system, man, artificial organs, medicine, environment. So if you see this, we have two branches, basically, these two green one. One is related to bioengineering, and the red one is artificial food. But when I start studying artificial food, I figure out that even artificial food itself is not enough, and you have to think about knee joint, hip joints, different kind of amputation, because we're dealing with what? With man, and he need all these different approaches from different point of view of this system. And the second branch is medicine. And I conditionally split in this diagram by three classes. Diagnostics. Sometimes they make amputation not on time, and we can prevent this. Medical drugs and therapy. So in parallel with this topic related to food, I start working with two systems. One related to bioengineering, and the second part related to pharmacology. Since we have many projects here, even for 1 hour, it's unreal to explain every project from three point of view. I just want to, in introduction, emphasize that we have been using all classical trees, all different methods we use. And at the beginning, I will tell in introduction that method we use, actually, this is classical trees, very extensive mathematical modeling, multiple synergism, and all achievement of modern science. We created few companies, and this is just two examples, Trees by Pharma International and Nigel LLC. And there are only pharma companies in the world that apply trees to all projects. There is no any project we do here in our corporation without trees. So the trees is an entry we use everywhere. Okay, by the way, Nigel, maybe I see the slide, we use a word legion because our medical drug like Roman legions, they fight different bacteria. It's from top to bottom. Now, so we use different roadmaps for all our projects. This is one example for bioengineering. We have all these projects, and where we in details go through all the stage up to final product. For analysis, we use the Ishikawa diagrams, for instance, for MDI resist, multi-drug resist bacteria. For all this project, we use also very nice approach for solving problems. Definitely system operator. For instance, this for pathogenic microorganism fighting. Only in our system operator, we also, since we work with nature, we included one more column, which is related to ideal final result. So this is for us is criteria. It's more convenient to work with, maybe unusual, but very convenient to work with. So any project we have these operators, past, present, future, as usual, classical. And this is screen, the same screen, but very important here that we go and we use law of evolutions, and we a lot of switch from macro to level, micro level, and vice versa in our research. So actually we use all law of evolutions. And I represented in this slide briefly that we have three branches. One is bioengineering with all some of our projects. This related not only prosthetics, orthotics, training device, electrostimulation, for scoliosis, etc. Drug design. Drug design I will split by two branches. One is repurposing drugs. What is it? Actually, it's generic drugs, which already known, but we, based on the synergy we provide and mathematical modeling, we use for each project silica modeling, mathematical modeling with neural links, and dynamic drugs. Dynamic drugs is a new, today I'm more concerned in this presentation about this is new generation of drugs of future. This is dynamic drugs. And definitely this is some examples for our project in diagnostic is by BCMM, blood self-morphometric method, cancer detection, atherosclerosis detection, etc. Always we have to think about the system. Not only you have to have drug, but also you have to know when to, how to apply. Here I represent tree structure of functional analysis method for improving the system man microbiome environment. I will not go in very detail of this, but we study system from different approach. And if you see, you can recognize in this stuff a lot of tools that we use in trees, trimming, for instance. And we study quasi-life system, which is very important in future. And this some name of these drugs in our corporation so far because they not become public as usually. So definitely we keep in mind development cycle of technical system. It's very similar to growth of microorganisms in nutrition medium. And according, everybody know this, all of us trees professionals, but the idea is more and more money included, for instance, spent in creating new kind of antibiotics, but unfortunately they don't work because system bacteria become resistant. So we're supposed to, if I had more options here, I would write another, I will represent another line, that this is a time for burning a new dynamic drugs. We'll see from here. This example of trimming, for instance. To visualize this trimming, for instance, we have standard multi-drug resistant approach. When if you have bacteria, we have different antibiotics. In our multi-drug resistant, we have just two of them, enhancers and antibiotic. Enhancer make less resistant this bacteria. Antibiotic, low dosage. This kill this bacteria. And this standard way, dosage extremely high right now. And this is a different approach. We have definitely we use all these trees principle. This is only one of examples. All apparatus with trees. And this is idea to survive rate of bacteria's innovations much faster than the rate of new antibacterial drugs development. This is time to find another non-traditional way to find multi-drug resistant bacteria. It's not related only one example, it's related to all our researches, because nature is more flexible than our static conservative drugs. So based on this, trees, just a second. Smart bacteria can be only conquered by smart method of treatment, not static anymore. So static also works. But if you look at this slide, and real efficacy of the drug is not advertised. Only statistically significant difference between the experimental groups and placebo advertised, statistical difference. If you look at this, this is because that efficacy of standard drugs for population rarely exceed even 50% if you see this, even low. For example, effectiveness of treatment with amlodipine reached 45%. And for people who treated this. We have many examples, for instance, treatment hepatitis C with combination of different interferon, etc. One of the reasons that people are different from each other, and they have different receptors. Drug and the virus in themselves, they not necessarily match their configuration of these receptors. And the drugs we use right now, they have conservative structure. Only 50% of people have a conservative drug that matches the receptor. And the rest have different receptors and do not match the drugs. For drugs to work for whole population, they must get the ability to adapt to a specific receptor and organism. They must be dynamic drugs, not conservative drugs with a clear known structure. This is few, I represented this in this slide, property of classical drug. Property of classical drugs, they have limit for efficacy in the population. Because they precise conservative chemical structures. They have slippage effect referred to the change in the person receptor apparatus over time. This can cause a loss of effectiveness in certain medications and substances such as AC inhibitors, which have been extensively studied. The drug cannot independently adapt to reduce the slippage effect or mutation in case of infections. It is uncommon for these drugs to have effectiveness rate above 50%, as not everyone has the same receptor apparatus as I mentioned before. Few words about property of dynamical drugs, if you have. Unlike classical drugs based on conservative chemical structure, what do I mean? I mean one molecule per drug, the same configuration molecule, I put this. Dynamic drugs have efficacy close to 100% in the population if they create it, because they will be effective for all people. A single polymer, such as protein, DNA, RNA, polysaccharides, motanin, can produce millions of derivatives within 1 mole of substance, despite having a complex and irregular structure. This is achieved by partially modifying the internal groups of the polymer, such as lysines, in the protein or amino groups in the DNA or RNA. Each structure is tailored to fit the receptor of a specific patient, making it one among many but uniquely suited to individual. Even there is mutation of polymorphism of receptor that cause a delay. The drug may still work. This is because there could be other variation of the same drugs, what were not effective earlier but can still be beneficial. When microorganisms and viruses mutate, drugs can still be effective because they have millions and millions of similar structures used for this formula, for instance, for combination with factorials, among which at least one can target the mutated version. This is because drugs contain predictive structure that can identify and target even not existing targets. Next slide is very demonstrative for this. Next slide. We name it. It represents the degree of freedom, of variability and adaptability. Pay attention to, we have different kind of circles. Every circle has different diameter in different area. And this slide shows a chart. The area is proportional to degree of freedom, variability and adaptability of different systems depending on external condition. If a condition will visualize the degree of variability of static classical drugs in human population, this is this blue circle. Then due to variability of microorganisms, fungi and viruses, the survival and degree variability of the latter overlap the variability of static drugs and force pharmaceutical company to throw billions of dollars into, I will say, into the wind, introducing new and new antibiotics. Look at this. This is actually classical conservative system. This is green one in human organism receptors. They can mutate, they can change configuration. That's we have slippage effect. If you see this gray circle, an area is bigger than this one, this is microorganism, because we came to microorganism. They exist million years. So their adaptability is much bigger. But if you create medical drugs, nowadays microorganisms, they are not strong enough. They don't expect this present for us. And if you look on this circle, which is represented by red, so you see area is much better, bigger, and future cell-adapted dynamic drugs and vaccine would be even bigger because we have more approach for this. This about this very demonstrative pharmacoid. And this approach makes possible to completely overcome the resistance of microorganisms, because our area of variability and adaptability much bigger than there due to such dynamic drugs. We work with quasi-life self-assembled drugs. And this slide, we represent 3 groups of drugs. We conditionally classify dynamic drugs. These are green agonists, dynamic version on interferon, for instance, interleukins, insulins, etc. Antagonist, this yellow, dynamic version of RNA or DNA, nucleotides. Okay, and finally, the next one is vaccine. I will not go in any depths a lot of this. Agonist dynamic drugs enhance some functions and exceed the activity of their original substance. For example, partially acylated alpha interferon is not only 10 to 50 times more active than native interferon, but also has a different, broader spectrum of action both on viruses and tumors. Antagonists are represented by gene therapy, agent based on antisense DNA of the RNA, representing a dynamic system of partially modified oligonucleotides. They can inactivate any number of genes and gene sequences for a period sufficient to trigger immunity and destroy the tumor. I will show in my presentation how it works. And eventually the third one, group vaccines, partially modified vaccine antigens proved effective way, even when administered orally. You know, usually vaccine we don't use orally, but in one of our graphs I'll show that even orally, its effectiveness compatible with injection. And when such as influenza vaccine was injected, it was effective in preventing a new strain of the virus. And this kind of vaccine had predictive property, protecting experimental animals from future mutation strains of the viruses. We have 2 branches in this one. This algorithm of creating classical drugs, this left branch, and dynamic drug, right part. Calculating the number of groups available for modification. We modify groups. I'll show how it works. In proteins, it's much easier than coming up with completely new molecule capable of stimulation or inhibiting their protein. So the great idea. Disadvantage of classic, as we mentioned. Classic, they have conservative chemical structures, the presence of slippage effect, change in receptor sensitivity in response to the same medication over time, diminish the loss of efficacy drugs over time, treating hypertension, diabetes, etc., multi-drug resistant, infection due to antibiotics, inability to adapt to new receptors, a new microorganism, defense factor, and mutation as a result, antibiotic function develop also slippage effect. Similar development tumor resistance to therapy over time observed in oncology related to chemotherapy drugs, also slippage effects. Everywhere we have slippage effect. Not enough flexibility. The slides show algorithm of creating agonist receptor similarity molecules. From X-ray bank, a known structure is taken, for instance, interferon, interleukins, etc. A structure is built by a program according to empirical data. The quality of lysine amino groups, histidine, serines, and threonines accessible for modification is calculated. We use special algorithms to calculate this accessibility. The necessary level of modification is calculated. They will provide for the maximum quantity of various derivatives from one volume of solution. As a result, there is increased activity of these modified proteins by a factor of 10 to 1,000 and more times. An increase in spectra and broader activity and effectiveness is population supposed to be close to 100% is adjustable for everybody. The activity increase is facilitating by changes the protein molecular charge to negative. That's key thing. So based on our approach, we change the protein molecular charge to negative, which increases its affinity to its own receptors. I will model what I said by this analogy. This is a great idea. Look at this. In this slide, you can see this is a key, standard key. Main idea behind the creation of dynamic drug is to use a set of lockpicks instead of one key for one lock, which represent the principle of classic drug with a conservative structure. A group of many similar molecules that open many locks and adapt to the target. This ensures almost 100% efficacy and the broadest possible spectrum of the drug action. Here we got many patents and publications about this and philosophy of the drug design for dynamic drugs. Subdividers. For instance, this acylation agent, they change the protein molecular charge. And there are agents for protein acylations. We have succinic anhydride, maleic anhydride, acetic anhydride, and aconitic anhydrides. We can say that in creation dynamic structure is incomplete, not completely, incomplete charge change to biopolymer molecules and polyacceptor molecules by acylating with D and 3 carboxylic acid anhydrides. The slide summarizes the most studied acylation agent, as I mentioned, this agent, which known for today. Okay. And this help to change positive charge of one amino group to one negative charge of the carboxyl group. Acetic anhydride, for instance, it does not reverse the charge, but neutralizes the positive charge of amino groups. It is highlighted in purple here. And aconite anhydride is highlighted red, this one. Changes one positive charge of amino groups to two negative charges of carboxyl groups. If more specifically, on the next slide, we have a change of molecular charge. Keep in mind, we go to micro level, right? Change of molecular charge to negative after acylation, succinylation, etc., based on all these anhydrides names, right? These nouns. And this positive charge, after, for instance, we apply succinic anhydride, we have some partly modified molecules which have negative charge. This is very important because you know negative and positive, they attract each other. And it charge proteins and lead to the creation of hundreds of thousands of proteins, hundreds of thousands and millions derivative from single molecule by just one reaction. In proteins, the amino groups of lysine and histidine residues and the hydroxyl groups of tyrosine, serine, and threonine undergo oscillations. As a result of such chemical reaction, proteins and other biopolymers acquire excess negative charge. At the same time, incomplete oscillation leads to formulation of hundreds of thousands of derivatives from one initial protein with one peptide core by different arrangements and permutation of carboxylic acid residues. Thus, dynamic supramolecular structure are formed. So this key thing of this technology. This one of a couple example of dynamic drugs. These two drugs that belongs to dynamic self-organized systems. So it took many years, really many years from idea to create this, and finally we did it. And this slide describes two drugs that belongs to dynamic self-organized system antagonism. For instance, albuvir. We name it like this. It's antiviral agent based on more than 1 million partially isolated peptides that can effectively block the nuclear import-export system for cell nucleus-dependent viruses. The efficacy of the drug has been confirmed for more than 25 volunteers and animal viruses. And this authorized, this drug is authorized for veterinary already in some countries. The second drug is FRNA. FRNA is more than 100,000 partially modified RNA oligonucleotides capable of selective accumulation in cancer cell, complementary interaction with cellular RNA, and stopping protein synthesis. This is very important because protein is source of life. The mechanism of FRNA action is similar to that of microRNA, but the resulting double helical hybrid is protected from the action of cell helicase enzymes, and the hybridization process does not depend on temperature and proceeds very quickly. So this is how it's solved. Examples, other examples of dynamic drugs. Two more. Dynamic insulin. Here it is. Has shown high biological activity when administered orally in rats with Alexander diabetes. We had groups of rats with this diabetes. And the system promoted reduction in glucose level to 10 millimole per liter on average, and maintained this level within 24 hours after a single application. It can be considered a candidate for development and implementation in capacity of oral insulin, since it's so active. Efficiency of the preparation was confirmed in animal by using both fasting and glucose load. Next example is dynamic antibiotics, very important. Instead of single antibiotic molecule, conservative structure, such as polymyxin. You know, polymyxin is last resort right now in drugs which can help you to fight the multi-drug resistant bacteria. Contains thousands of derivatives which incompletely substitute hydroxyl and amino groups. They have a broader spectrum of action and are effective against multi-drug resistant bacteria. So a few words about, we made animation how albuvir has ability change or adapt to different viruses and known as dynamic antiviral drug. For instance, here we see animation of mechanism of viral genome penetration through the cell membrane. On the surface of the cell membrane are nuclear pores containing receptors for alpha and beta importins. Viruses themselves contains importin-like protein and are able to activate receptors on the cell nucleus, open nuclear pores, and penetrate into nuclei. And you see how he easily penetrates to the nuclei. But let's apply albuvir. Albuvir represented by this red. This slide summarizes the mechanism of action of albuvir. Albuvir is modified peptides that block viral impортins and prevent the viral genome from penetration the cell nucleus. Albuvir has no immunomodularity or immunotropic properties, but its effect in influenza is amazing. Can you believe? It stops replication for any strains on influenza in 30 minutes. Albuvir is currently authorized to use an antiviral veterinary drugs for therapy of the widest range of veterinary viral infections. And see how it works. This is our albuvir, the red one. And it blocks. No key anymore. We have set of publication and patents for this in different countries. Okay. Next example. I want to go to say a few words about dynamic anti-cancer drug is medication that effective in fighting cancer cell and can be adapted to different types of cancer. A few words about this. There are a lot of anti-cancer drugs on the market, including both direct cytotoxic agent, anti-cancer, cytostatic, and various types of immunomodulators. The use of various combination of these drugs usually does not lead to complete cure of the most cancers. Current anti-cancer drug, despite clinical efficacy, also cause numerous serious side effects. Anti-cancer cytostatic drugs, side effects: nausea, vomiting, hair loss, this bone marrow suppression. Very often the market efficacy of chemotherapy in the initial stage of treatment is reduced to ineffectiveness during long-term treatment. Especially distant cells for metastasis become practically insensitive to therapy due to the effect of selection of resistant cells. This type selection effect is also typical for microorganisms and viruses. The more poisonous and toxic substances to the tumor are invented by scientists, the faster the cancer cell adapt to the therapy. Sometimes it occurs even right after the first application. So, slippage effect. To solve the above problems, several difficulties had been solved. It was necessary for the drug to be attracted and accumulate mainly in cancer cell, not in all cells, cancer cell, because we fight with cancer. Suppress cancer cell growth, cancer cell, not all cells, and the selective, right? And the drug should not be toxic to healthy cells of the host body. The ideal candidate for anti-cancer drug should be chosen from existing drug produced by pharmaceutical companies. In our approach, not necessarily we have two ways, either create new molecules or use molecules, but make it smart. We propose to target the protein synthesis because protein is source of life in cancer cells. It is well known that cancer cells have a unique ability to increase pinocytosis and phagocytosis, as well as selectively capture RNA and DNA oligonucleotides. Very interesting how we did it here. This action mechanism of antisense polymorphic RNA, FRNA. After temperature denaturation of DNA, and this is the first step, both chains are acylated with a modifier that reverses the charge of a mononucleotide. Such chains have the ability to selectively hybridize only with their precursors. For example, chain A will, no, for instance, this chain A here, right? Can selectively hybridize only with isolated chain A of them. And the same chain B, this, selectively hybridize only with the isolated chain B. And as a result, we get this combination. And we have, could be more and more this configuration of this. Okay, just a second. Somebody sent me a message, right? About, let me save it. English? No, I believe answer question, right? This slide shows the principle of hybridization of modified nucleotides with their precursors. In particular, when the charge is reversed, adenine in the DNA and RNA structure hybridized when acyl adenine, guanine, acylguanine, and cytosine with acylcytosine. At the same time, okay, we see this hybridization. The principle of bond formation changes from hydrogen bonding, very important, to hydrogen and ionic bonding. As a result, specific double helical polynucleotide stable to the action of helicase and are formed here. The principle of obtaining the anti-cancer drug nucleamide-based antisense RNA. For instance, we have yeast, right? Transport RNA, such as alanine tRNA, transport RNA. It is taken and treated with nuclease, which lead to the formation of multiple oligonucleotides, and then modified by acylation to reverse the charge of oligonucleotides residues. As a result, definitely we skip many intermediate steps. It's just roughly what we have here for this process to get dynamic RNA. Such carboxylated fragments, I show in previous slides, and we have here. Most cancer cells have the ability to capture oligometric RNA fragments by pinocytosis. Normal cells, on the other hand, utilize little or no pinocytosis. The fRNA accumulate many in cancer cells. They specifically hybridized with alanine tRNA, which leads to inactivation of protein synthesis on polyribosomes and apoptosis of the cancer cells. You know, apoptosis, dying. The number of RNA fragments initially exceed very excessive. It is dynamic structure that self-assembly on tRNA. It is very difficult for a cancer cell to adapt to such therapy. Actually, we solve the problem of selective using this, our dynamic drugs. And in next slide, you will see how polyribosomes and mRNA mechanism in action here. The slide shows animation of mechanism of protein translation blockage during hybridization of mRNA fragment with mRNA. The same principle is for transport RNA, applies to matrix RNA, which can also be disrupted by formation of supramolecular complexes we go to macro level with modified fragments of the original matrix RNA. mRNA, it's messenger RNA or mRNA, is genetic material that tells your body how to make protein. And let's see how it works. So these fragments of our RNA, right? Cancer cell with pinocytosis selectively snatch oligometric RNA fragments with altered charge of RNA from the intercellular space and die due to blockage of protein synthesis. So the regular cells do not get, right, non-cancerous cells. Imagine this is, I made example with, you will see next slide. The jaw, maybe you remember the small jaws. And since no protein synthesis, they become shrinking, selectively cancer cells, not normal cells. This is idea of our, we name it antican between us. This slide shows the result of experimental study of several anti-cancer drugs and in immunomodulatory lectin from a bacterium. The FRNA and its combination with immunomodulatory vaccine show the best result. At 40 days after inoculation of Lewis carcinoma mice, only the group receiving FRNA and lectin survived all animals. And you can see this black. The rest of them, they pass away. Black squares, right? This one.
Dr. Farber, you have 10 minutes for your speech and question session.
Thank you. Thank you for telling. So I try to do briefly. And this is a slide show how FRNA influences the dynamic of tumor growth. I will briefly go. We have tons of different publications and patents for this. And this is dynamic vaccine. A few words about dynamic vaccine. So the slide shows the methodology of dynamic vaccine and why they are effective when administered orally. If antigen is partially modified, thousands of antigens with slightly altered surface architectonics are formed. When administered orally, enzymes will only degrade unmodified antigens, and this will be no immune response, while modified antigens will be protected and is degraded to immunogenetic haptens that can be absorbed into bloodstream and induced to immune response. So I skip some slides here. Okay, and show. This is strategy of dynamic for creating dynamic vaccine based on self-organized modified peptide. We took classical syngnoic and Newcastle disease viruses antigen from commercial vaccine and make it modification. And the next slide, look at this, very interesting slide. So on this x-axis, we have level of oscillation. Remember, we use oscillation method. And vertical line, antibody titer. And this is, usually we have infection, right? Usually we have injection vaccine. And this injection vaccine. And level of oscillation, what is 0? 0 is regular vaccine, no implementation of our oscillation method. But we have, when we have approximately 3%, you see even the same vaccine efficacy increase more than 4 times here. The same vaccine. Moreover, if you look at this yellow curve, so it works even orally. A result orally compatible to normal vaccine when it use injection. So this is absolutely interesting approach. I skip this. We also have many patterns for this. And this is dynamic parallel insulin, very important because many people, they suffer this, use insulin every time injection, etc. And I skip this idea since we have limited time. And we move here also effects of dynamic insulin with afterload glucose level in blood of rats with Alexander diabetes. And after administration of 1 hour, you see this, of dynamic insulin, the blood glucose level in rats particularly returns to normal. Whereas untreated control remains high. There is some of our publication. Dynamic antibiotics. So we use a lot of mathematical modeling, and this is some example where we study schematic diagram of chemical reaction of dynamization of antibiotic polymyxin. Polymyxin has 7 amino groups available for modification. Not every drug can be modified. We use silica modeling to detect this. To dynamize its structure, not all groups need be substituted, but only a certain calculated number. And how we calculate, we use combinatorical. This is a result of this formula. As a result, for instance, about 20,000, 16,000 something different arrangement and combinatoric of substituents are formed from one polymyxin. In addition, the former product react through ionic bonds, which each other to form a complex supramolecular assemble.
Dr. Farber.
Yes.
Can we start our session question session because we have only 5 minutes?
Sure. Welcome.
Okay.
So I just summarized that.
Okay.
Let me skip everything, right? I skip this one, only say, okay. Just want to show that there are a lot of different applications of this. We have also, I just name of this, dynamic hemostatic drug gamma, which tremendously stop blood bleeding, right? This, right? Have many of them. And summarization, optimization of dipyrimidamole. Dipyridamole is would be for wound we use it, but also can be used for cosmetology. Fantastic result is shown. Okay, also a result of this chromatograph research. So all this stuff. And finally, we applied this method for not only for drugs, all these apparatus, 3 apparatus, 2 different inventions. And I made presentation for 500 years of Leonardo da Vinci death in Florence about all this stuff. June 16, the day of this, and today is important day. Today is 97 years old, and happy birthday for our trees created Genrich Schröder and this question and answer session, this contact. Yes. So I finish.